All right. Hey, everybody. We're going to carry on with our talk about enzymes. And so last time we talked about, we reviewed and went into a little bit more detail about the properties of proteins. We talked about amino acid structure. We talked about amino acid R groups. And we talked about the levels of protein structure. And so we talked really, one of the things that we left with, the things that we ended with, was this idea that if you change the primary structure of a protein, then you change the shape of the protein. And if you change the shape of the protein, then you change the function of the protein. And so if we mess with these bonds, either the hydrogen bonding or the hydrophobic interactions or the disulfide bridges or the ionic bonds, if we mess with that, then what can happen is something called denaturation, which really is just a fancy way for saying unfolding. So this just means unfolding. Okay, or losing the 3D shape. Okay, so if we lose the 3D shape, then that's called denaturation. And so we can cause the protein to change its shape by changing the primary sequence of the polypeptide chain, but we can also cause it to change shape or unfold if we change the environment that it's in, especially the temperature and the pH. Proteins are really, really sensitive to changes in temperature or in pH. So they need to have a very specific range of temperature and pH. Okay, and so these things together, these things are called the optimal range. You could say the optimal temperature range. Okay, or you could say the optimal pH range. And if I do this, that just means it's the same as whatever's above, right? So this just means optimal pH range. And notice that it's a range of temperatures, and it's a range of pHs. What does that mean? It means that there are more. It's like kind of like a space of values. So and just to go back to this here, optimal. What does optimal mean? Optimal means the best, right? Or the favorite. So if it's the best range, then let's say that, and so for example, human temperature. There's optimal human temperature, and it's not, a lot of times we say, oh, well, it's 37 degrees, but that's not actually true. Sure, 37 is the average, so this is like the average temperature, but really the optimal range is anywhere from like 36 to 38. Anywhere in there is fine. At 38, you're feeling a little icky. At 36, you're feeling a little bit chilly. But in this range here, is the optimal range for most proteins in the human body. Now, if you move outside of that range, then that becomes not very optimal. So, that can, in some cases, that can cause the protein to become unfolded. And when it becomes unfolded, then it loses its shape, right? So if it unfolds, then it loses its 3D shape. And therefore, its function, or I guess it decreases its function. right? So that's not good. And if you go far enough, if it loses enough of its shape, if it fully denatures, I guess this should say fully, let's not do that. Let's just add to this. Okay, it's partially denatured would equal to decreased function. So it just doesn't function as well if it's partially denatured. But if it's fully denatured, if it denatures all the way back to its polypeptide sequence, all the way back to its primary structure, then it will be non-functional. In other words, it completely loses its function. And typically, once a protein has denatured, once it's unfolded, it cannot return to its functional form. So there's processes in the cell that, that help with this. But yeah, you have to understand that if we denature a protein, it's toast. It's done. It needs to just be recycled, essentially. It needs to be recycled for amino acids. Okay. Now a little bit more about optimal range. So now we've got two organisms here. So we've got this guy right here, this crazy looking bacteria, and we've got this nice happy looking bacteria. And you can see that they come from two different environments. So this environment over here, this environment right here, is a thermal hot spring. This is a thermal hot spring, which means that it's very hot, right? It's very hot. And it's also, you can see all these different colors here represent different chemicals. So it's got lots of chemicals in there. So it's pH. The pH is probably some sort of extreme. So we could say that it's got some extreme pH. Okay, but over here, look at this nice place. This place here, nice. The temperature here is nice and moderate, right? It's not too hot, not too cold. The pH is, you know, somewhere, let's call it somewhat neutral. It might be neutral, I don't know. But it feels like it's probably not too acidic, not too alkaline, right? It's fairly like neutral pH. And so they live in very different habitats. So the proteins that exist in this organism are going to have a different optimum range than the proteins that exist in this organism. And that's because the environment that they live in is really going to determine the optimal range for the proteins in those organisms. So, yeah, I guess that we just want to make sure that we understand that habitat, or the environment, I guess we could say, environment. Sorry, that's really horrendous. Environment. Okay, or the organ system and its intended function. So for example, the cells of the stomach, right? So in the stomach, we'll talk about this a little bit. Stomach has a very low pH, and we'll talk about that in a minute. Okay, so if the temperature in this hot spring is 80 degrees Celsius and it has a low pH of 3, it's very acidic, right? Well, then the enzymes that exist in this organism here also need to have a pH and a temperature, an optimal range, and an optimal temperature range and an optimal pH range that's somewhere around there, right? So I would say that like the temperature range might be somewhere, let's say that it's, I don't know, 60 to, I don't know, even 100 degrees C, or 95 degrees C is the optimal range for the temperature the optimal temperature range for the proteins in that organism. And then the optimal pH range is somewhere like between 2 and 4 for the proteins in that organism. Now, this organism over here that's living in these nice, fairly what we would think of as normal conditions, well, the proteins in this organism would have an optimal range where 15 degrees Celsius is probably within that optimal range, right? So maybe the optimal range is 10 to, let's say, 30 degrees Celsius. And the optimal pH is going to be somewhere around 6.5, right? So maybe it's between 5 and 7 is the pH, the optimal pH for this guy here, right? Maybe it's just like slightly acidic. Okay, so there's going to be an optimal range here. And it's going to reflect the environment that you find them in. Okay. Anything else we want to say about that? All right, now we're getting into some different stuff here. All right. Let's end it there. So next time what we'll do is we'll talk about chemical reactions and just how chemical reactions help with, or how they relate to enzymes. But for now, we'll just kind of end it with optimal range.